So hello everyone and welcome back to Neetopedia, that is Neetka Encyclopedia, and we are back again with our biology audiobooks of plant physiology, second chapter, that is mineral nutrition. Now before starting, please note that our next video will also be on mineral nutrition as well, about the tips, tricks and mnemonics to memorize the different concepts of this chapter. So subscribe to the channel, hit the bell icon and stay tuned with us for further updates. Now without wasting our time, let's begin with Chapter 12, Mineral Nutrition. The basic needs of all living organisms are essentially the same. They require macromolecules such as carbohydrates, proteins and fats, and water and minerals for the growth and development. This chapter focuses mainly on the inorganic plant nutrition, wherein you will study the methods to identify elements essential to growth and development of plants and the criteria for establishing the essentiality. You will also study the role of essential elements, their major deficiency symptoms, and mechanism of absorption of these essential elements. The chapter also introduces you briefly to the significance and mechanism of biological nitrogen fixation. 12.1 Methods to study the mineral requirements of plants. In 1860, Julius von Zach, a prominent German botanist, demonstrated it for the first time, that plants could be grown to maturity in a defined nutrient solution in complete absence of soil. This technique of growing plants in nutrient solution is known as hydroponics. Since then, a number of improvised methods have been employed to try and determine the mineral nutrients essential for plants. The essence of all these methods involves the culture of plants in soil-free, defined mineral solution. These methods require purified water and mineral nutrient salts. Can you explain why this is so essential? After a series of experiments in which the roots of the plants were immersed in nutrient solution and wherein any element was added, substituted or removed or given in varied condition, a mineral solution suitable for the plant growth was obtained. By this method, essential elements were identified and the deficiency symptoms were discovered. Hydroponics has been successfully employed as a technique for the commercial production of vegetables such as tomato, seedless cucumber and lettuce. It must be emphasized that nutrient solutions must be adequately aerated to obtain the optimum growth. What would happen if solutions were poorly aerated? Diagrammatic views of hydroponic techniques is given in figure 12.1 and 12.2. 12.2 Essential mineral elements. Most of the minerals present in soil can enter plants through roots. In fact, more than 60 elements of the 105 discovered so far are found in different plants. Some plant species accumulate selenium, some others gold, while some other plants growing near nuclear test site take up radioactive strontium. There are techniques that are able to detect the minerals at even a very low concentration, that is 10 to the power minus 8 gram per milliliter. The question is whether all the diverse mineral elements present in plants, for example, gold and selenium as mentioned above, are really necessary for plants? How do we decide that what is essential for plants and what is not? 12.2.1 Criteria for essentiality. The criteria for essentiality of an element are given below. A. The element must be absolutely necessary for supporting normal growth and reproduction. In the absence of the element, the plants do not complete their life cycle or set the seeds. B. The requirement for the element must be specific and not replaceable by other element. In other words, deficiency of any one element cannot be met by supplying some other element. C. The element must be directly involved in metabolism of the plant. Based upon the above criteria, only a few elements have been found to be absolutely essential for plant growth and metabolism. These elements are further divided into two broad categories based on their quantitative requirements: macronutrients and micronutrients. Macronutrients are generally present in plant tissues in large amount, that is in excess of 10 millimole per kg of dry matter. The macronutrients include carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium and magnesium. Of these, carbon, hydrogen and oxygen are mainly obtained from CO2 and water, while others are absorbed from the soil as mineral nutrition. Micronutrients or trace elements are needed in very small amounts, less than 10 millimole per kg of dry matter. These include iron, manganese, copper, molybdenum, zinc, boron, chlorine and nickel. In addition to the 17 essential elements named above, there are some beneficial elements such as sodium, silicon, cobalt, and selenium. They are required by higher plants. Essential elements can also be grouped into 4 broad categories on the basis of their diverse functions. These categories are: number 1, essential elements as components of biomolecules and hence structural elements of cells. For example, carbon, hydrogen, oxygen, and nitrogen. Number 2, essential elements that are components of energy-related chemical compounds in plants. For example, magnesium in chlorophyll, phosphorus in ATP. Number 3, essential elements that activate or inhibit enzymes. For example, Mg2+ is an activator for both ribulose bisphosphate carboxylase oxygenase and phosphoenol pyruvate carboxylase, that is Rubisco and pepcase respectively, both of which are critical enzymes in photosynthetic carbon fixation. Zn+2 is an activator of alcohol dehydrogenase and MO of nitrogenase during metabolism of nitrogen. Can you name a few more elements that fall in this category? For this, you will need to recollect some of the biochemical pathways you have studied earlier. Number 4, some essential elements that can alter the osmotic potential of a cell. Potassium plays an important role in the opening and closing of stomata. You may recall the role of minerals as solutes in determining the water potential of a cell. 12.2.2 Role of macro and micronutrients. Essential elements perform several functions. They participate in various metabolic processes in plant cells such as permeability of a cell membrane, maintenance of osmotic potential of cell sap, electron transport systems, buffering action, enzymatic activity, and act as major constituent of macromolecules and coenzymes. Various forms and functions of essential nutrient elements are given below. Nitrogen. This is the essential nutrient element required by plants in the greatest amount. It is absorbed mainly as nitrate, though some are also taken as nitrite or ammonium ion. Nitrogen is required by all parts of a plant, particularly the meristematic tissues and the metabolically active cells. Nitrogen is one of the major constituents of proteins, nucleic acids, vitamins, and hormones. Phosphorus. Phosphorus is absorbed by the plant from soil in the form of phosphate ions, either as H2PO4- or HPO4-2-. Phosphorus is a constituent of cell membranes, certain proteins, all nucleic acids, and nucleotides and is required for all phosphorylation reactions. Potassium. It is absorbed as potassium ion. In plants, this is required in more abundant quantities in meristematic tissues, buds, leaves, and root tips. Potassium helps to maintain an anion-cation balance in cells and is involved in protein synthesis, opening and closing of stomata, activation of enzymes in the maintenance of the turgidity of cells. Calcium. Plants absorb calcium from the soil in the form of calcium ions. Calcium is required by meristematic and differentiating tissues. During cell division, it is used in synthesis of cell wall, particularly as calcium pectate in middle lamella. It is also needed during the formation of mitotic spindle. It accumulates in older leaves. It is involved in normal functioning of the cell membranes. It activates certain enzymes and plays an important role in regulating metabolic activities. Magnesium. It is absorbed by the plants in the form of divalent Mg2+ ion. It activates the enzymes of respiration, photosynthesis, and are involved in the synthesis of DNA and RNA. Magnesium is a constituent of the ring structure of chlorophyll and helps to maintain the ribosome structure. Sulfur. Plants obtain sulfur in the form of sulfate ion. Sulfur is present in 2 amino acids, cysteine and methionine, and is the main constituent of several coenzymes, vitamins such as thiamine, biotin, coenzyme A, and ferredoxine. Iron. Plants obtain iron in the form of ferric ions Fe3+. It is required in larger amounts in comparison to the other micronutrients. It is an important constituent of protein involved in transfer of electrons like ferredoxine and cytochromes. It is reversibly oxidized from Fe2+ to Fe3+ during electron transfer. It activates catalase enzyme and is essential for the formation of chlorophyll. Manganese. It is absorbed in the form of manganese ion, that is Mn2+. It activates many enzymes involved in photosynthesis, respiration, and nitrogen metabolism. The best defined function of manganese is the splitting of water to liberate oxygen during photosynthesis. Zinc. Plants obtain zinc as Zn+2 ions. It activates various enzymes, especially carboxylases. It is also needed in the synthesis of oxine. Copper. It is absorbed as cupric ions, that is Cu2+. It is essential for overall metabolism in plants. Like iron, it is associated with certain enzymes involved in redox reaction and is reversibly oxidized from Cu+ to Cu2+. Boron. It is absorbed as BO33- or BO4072-. Boron is required for uptake and utilization of Ca2+. Membrane functioning, pollen germination, cell elongation, cell differentiation, and carbohydrate translocation. Molybdenum. Plants obtain it in the form of molybdate ions, that is O2+. It is a component of several enzymes including nitrogenase and nitroreductase, both of which participate in nitrogen metabolism. Chlorine. It is absorbed in the form of chloride ion along with sodium and potassium ions. It helps in determining the solute concentration and the anion-cation balance in cells. It is essential for water splitting reaction in photosynthesis, a reaction that leads to oxygen evolution. 12.2.3 Deficiency symptoms of essential elements. Wherever the supply of an essential element becomes limited, plant growth is retarded. The concentration of the essential element below which the plant growth is retarded is termed as critical concentration. The element is said to be deficient when present below the critical concentration. Since each element has one or more specific structural or functional role in plants, in the absence of any particular element, plants show certain morphological changes. These morphological changes are indicative of certain element deficiencies and are called deficiency symptoms. The deficiency symptoms vary from element to element and they disappear when the deficient mineral nutrient is provided to the plant. However, if deprivation continues, it may eventually lead to the death of the plant. The parts of the plant that show deficiency symptoms also depend upon the mobility of the element in the plant. For elements that have actively mobilized within the plant and exported to the young developing tissues, the deficiency symptoms tend to appear first in the older leaves. For example, the deficiency symptoms of nitrogen, potassium, and magnesium are visible first in the senescent leaves. In the older leaves, biomolecules containing these elements are broken down, making these elements available for mobilizing to younger leaves. The deficiency symptoms tend to appear first in the young leaves whenever the element is relatively immobile and are not transported out of the mature organs. For example, elements like sulfur and calcium are a part of structural component of the cell and hence are not easily released. This aspect of mineral nutrition of plants is of great significance and importance to agriculture and horticulture. The kind of deficiency symptoms shown in plants include chlorosis, necrosis, stunted plant growth, premature fall of leaves and buds, and inhibition of cell division. Chlorosis is the loss of chlorophyll leading to yellowing in leaves. This symptom is caused by a deficiency of elements like nitrogen, potassium, magnesium, sulfur, iron, manganese, zinc, and molybdenum. Likewise, necrosis or death of tissues, particularly leaf tissue, is due to deficiency of calcium, manganese, copper, potassium. Lack or low level of nitrogen, potassium, sulfur, and molybdenum causes inhibition of cell division. Some elements like nitrogen, sulfur, molybdenum delay flowering in the concentration in plant and low. Please note that the tips and tricks to memorize these diseases and the symptoms caused due to various elements will be uploaded on a Telegram channel. If you haven't joined the Telegram channel till now, the link is in the description. Please join it soon. You can see from the above that the deficiency of any element can cause multiple symptoms and that the same symptoms may be caused by the deficiency of one of several different elements. Hence, to identify the deficient element, one has to study all the symptoms developed in all the various parts of the plant and compare them with the available standard tables. We must also be aware that different plants also respond differently to the deficiency of the same element. 12.2.4 Toxicity of micronutrients. The requirement of micronutrients is always in low amounts, while the moderate decrease causes the deficiency symptoms and a moderate increase causes toxicity. In other words, There is a narrow range of concentration in which the elements are optimum. Any mineral ion concentration in tissues that reduces the dry weight of tissues by about 10% is considered toxic. Such critical concentrations vary widely among different micronutrients. The toxicity symptoms are difficult to identify. Toxicity levels for any element also vary for different plants. Many a times, excess of an element may inhibit the uptake of another element. For example, the spots surrounded by chlorotic veins. It is important to note that manganese competes with iron and magnesium for uptake, with manganese for binding with enzymes. Manganese also inhibits calcium translocation in shoot apex. Therefore, excess of manganese may in fact cause deficiencies of iron, magnesium, and calcium. Thus, what appears as the symptoms of manganese toxicity may actually be the deficiency symptoms of iron, magnesium, and calcium. Can this knowledge be of some importance to a farmer, a gardener, or even for you in your kitchen garden? 12.3 Mechanism of absorption of elements. Much of the studies on mechanism of absorption of elements by plants has been carried out in isolated cells, organs, or tissues. These studies revealed that the process of absorption can be demarcated into two main phases. The first phase, an initial rapid uptake of iron into free space or the outer space of the cells. The apoplast is passive. And the second phase of uptake, the ions are taken in slowly into the inner space, the symplast of the cells. The passive movement of ions into the apoplast usually occurs through ion channels, the transmembrane proteins that function as selective pores. On the other hand, the entry or exit of ions into and from the symplast requires the expenditure of metabolic energy. Such is an active process. The movement of ions is usually called flux. The inward movement into the cell is influx and the outward movement is efflux. You have read the aspects of mineral nutrient uptake and translocation in plants in chapter 11. 12.4 Translocation of solutes. Mineral salts are translocated through xylem along with the ascending stream of water, which is pulled up through the plant by transpirational pull. Analysis of xylem sap shows the presence of mineral salts in it. Use of radioisotopes, mineral elements also substantiate the view that they are transported through the xylem. You have already discussed the movement of water in xylem in chapter 11. 12.5 Soil as reservoir of essential elements. Majority of nutrients are essential for growth and development of plants become available to the roots due to weathering and breakdown of rocks. These processes enrich the soil with dissolved ions and inorganic salts. Since they are derived from the rock minerals, their role in plant nutrition is referred to as mineral nutrition. Soil consists of varieties of substances. Soil not only supplies minerals but also harbors nitrogen-fixing bacteria. Other microbes hold water, supplies air to the roots, and act as matrix that stabilizes the plant. Since deficiency of essential minerals affect the crop yield, there is often a need for supplying them through fertilizers. Both macronutrients and micronutrients form components of fertilizers and are applied as per the need. 12.6 Metabolism of nitrogen. 12.6.1 Nitrogen cycle. Apart from carbon, hydrogen, and oxygen, nitrogen is the most prevalent element in living organisms. Nitrogen is a constituent of amino acids, proteins, hormones, chlorophyll, and many of the vitamins. Plants compete with microbes for the limited nitrogen that is available in the soil. Thus, nitrogen is a limiting nutrient for both natural and agricultural ecosystems. Nitrogen exists as two nitrogen atoms joined by very strong triple covalent bond. The process of conversion of nitrogen to ammonia is termed as nitrogen fixation. In nature, lightning and ultraviolet radiation provide enough energy to convert nitrogen into nitrogen oxides like NO, NO2, N2O. Industrial combustions, forest fires, automobile exhaust, and power generating stations are also the sources of atmospheric nitrogen oxides. Decomposition of organic nitrogen into dead plants and animals into ammonia is called ammonification. Some of this ammonia volatilizes and re-enters the atmosphere, but most of it is converted into nitrate by soil bacteria in the following steps. Ammonia is the first oxidized to nitrite by the bacteria Nitrosomonas and or Nitrococcus. The nitrite is further oxidized to nitrate with the help of bacteria Nitrobacter. These steps are called nitrification. These nitrifying bacteria are chemoautotrophs. The nitrate thus formed is absorbed by plants and is transported to the leaves. In leaves, it is reduced to form ammonia that finally forms the amine group of amino acids. Nitrate present in the soil are also reduced to nitrogen by the process of denitrification. Denitrification is carried by the bacteria Pseudomonas and Theobacillus. 12.6.2 Biological nitrogen fixation. Very few living organisms can utilize the nitrogen in the form of N2. Available abundantly in air, only certain prokaryotic species are capable of fixing nitrogen. Reduction of nitrogen to ammonia by living organism is called biological nitrogen fixation. The enzyme nitrogenase, which is capable of nitrogen reduction, is present exclusively in prokaryotes. Such microbes are called N2 fixers. Nitrogen fixing microbes could be free living or symbiotic. Examples of free living nitrogen fixing aerobic microbes are Azotobacter and Bigenicia, while Rhodosphaerium is anaerobic and free living. In addition, a number of cyanobacteria such as Anabina and Nostoc are also free living nitrogen fixers. Symbiotic biological nitrogen fixation. Several types of symbiotic biological nitrogen fixation associations are known. The most prominent among them is legume bacterial relationship. Species of rod-shaped rhizobium such as relationship with the roots of several legumes such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover beans, etc. The most common association on root is as nodules. These nodules are small outgrowth on the roots. The microbe Frankia also produces nitrogen-fixing nodules on the roots of non-leguminous plants and lists. Both rhizobium and Frankia are free living in soil, but as symbionts can fix atmospheric nitrogen. Uproot any one plant of a common pulse just before flowering. You will see near spherical outgrowths on the roots. These are nodules. If you cut through them, you will notice the central portion is red or pink. What makes nodules pink? This is due to the presence of leguminous hemoglobin or leg hemoglobin. Nodule formation. Nodule formation involves the sequence of multiple interactions between rhizobium and roots of the host plant. Principal stages in the nodule formation are summarized as follows. Rhizobia multiply and colonize the surroundings of the root and get attached to the epidermal and root hair cells. The root hairs curl and the bacteria invade into the root hair. An infection thread is produced carrying the bacteria into the cortex of the root, where they initiate the nodule formation in the cortex of the root. When the bacteria are released from the thread into the cells, which leads to differentiation and specialization of nitrogen-fixing bacteria. The nodule thus formed establishes a direct vascular connection with the host for exchange of nutrients. These events are depicted in figure 12.4. The nodule contains all the necessary biochemical components such as the enzyme nitrogenase and leg hemoglobin. The enzyme nitrogenase is a MOFE protein that catalyzes the conversion of atmospheric nitrogen to ammonia, the first stable product of nitrogen fixation. The reaction is as follows. N2 plus 8 electron plus 8 proton plus 16 ATP gives 2 ammonia plus H2 plus 16 ADP plus 16 inorganic phosphate. The enzyme nitrogenase is highly sensitive to the molecular oxygen. It requires anaerobic conditions. The nodules have adaptations that ensure that enzyme is protected from oxygen. To protect these enzymes, the nodule contains an oxygen scavenger called leg hemoglobin. It is interesting to note that these microbes live as aerobes under free-living conditions when nitrogenase is not operational, but during nitrogen-fixing events, they have anaerobic, thus protecting the nitrogenase enzyme. You must have noticed that in our reaction, ammonia is synthesized when nitrogenase requires a very high input of energy, that is 8 ATP for each ammonia produced. The energy required thus is obtained from the respiration of the host cells. Fate of ammonia. At physiological pH, ammonia is protonated to form ammonium ion, while most of the plant can assimilate nitrite as well as ammonium ions. The latter is quite toxic to the plants and hence cannot accumulate in them. Let us now see how ammonium ion is used to synthesize amino acids in plants. There are two main ways in which it can take place. Number 1, reactive amination. In this process, ammonia reacts with alpha-ketoglutarate acid and forms glutamic acid as indicated in the equation given below. Number 2, transamination. It involves the transfer of amino group from one amino acid of the keto group to a keto acid. Glutamic acid is the main amino acid from which the transfer of NH2, the amino group, takes place, and other amino acids are formed through transamination. The enzyme transaminase catalyzes all such reactions. The two most important amides, asparagine and glutamine, found in plants are structural parts of proteins. They are formed from two amino acids, namely aspartic acid and glutamic acid, respectively. By addition of another amino group to each, the hydroxyl part of the acid is replaced by another NH2- radical. Since amides contain more nitrogen than the amino acid, they are transported to other parts of the plant via xylem vessels. In addition, along with the transpiration stream, the nodules of some plants, for example soya bean, export fixed nitrogen as ureates. These compounds also have particularly high nitrogen is to carbon ratio. Summary. Plants obtain their inorganic nutrients from air, water, and soil. Plants absorb a wide variety of mineral nutrients or elements. Not all mineral elements what they absorb are required by plant. Out of more than 105 elements discovered so far, less than 21 are essential and beneficial for normal plant growth and development. The elements required in large quantities are called macronutrients, while those required in less quantities or in trace are termed as micronutrients. These elements are either essential constituents of proteins, carbohydrates, fat, nucleic acid, etc., or take part in various metabolic processes. Deficiency of each of these essential elements may lead to symptoms called deficiency symptoms. Fluorosis, necrosis, stunted growth, impaired cell division, etc. are some prominent deficiency symptoms. Plants absorb minerals through roots either by passive or active processes. They are carried to all parts of the organism through xylem along with water transport. Nitrogen is very essential for sustenance of life. Plant cannot use atmospheric nitrogen directly, but some of the plants in association with nitrogen-fixing bacteria, especially roots of legumes, can fix this atmospheric nitrogen into biologically usable forms. Nitrogen fixation requires strong reducing agent and energy in the form of ATP. Nitrogen fixation is accomplished with the help of nitrogen-fixing microbes, mainly rhizobium. The enzyme nitrogenase, which plays an important role in biological nitrogen fixation, is very sensitive to oxygen. Most of the processes take place in anaerobic environment. The energy ATP required is provided by the respiration of host cells. Ammonia produced following nitrogen fixation is incorporated into amino acids as amino group. So that's the end of the chapter. That's it for today. See you again in the next one. But till then, subscribe to the channel and stay tuned. Thank you.